Hello everyone and welcome. Good morning, good afternoon, good evening. My name is Carmen Guguta. I'm your usual host from Technobis Crystallization Systems, and today's webinar, Clinical Phases 1 and 2, the challenges for API crystallization processes, is brought to you today by Dr. Sophie Janbon from AstraZeneca. I hope you really see my slides. Sophie, can you confirm to me that, because I have on the monitor, I see something else. Okay.
Yes.
Okay, super. Thank you very much, Sophie. Just before we start, a bit of technical information to share with you. The webinar is being recorded and will be made available to you most probably tomorrow. You will receive an email where you will find exactly the link where you can rewatch the webinar or share it with other colleagues or viewers, the same as I think the PDF of the slides of Sophie. Although I didn't ask Sophie if she's willing to share them, she will tell us shortly. If you have any questions, please write to us making use of the chat function or the questions here in this platform, or send us an email at info@crystallizationsystems.com. The same in the coming days. If something pops up, just reach out to us and we will make sure your questions reach Sophie. The webinar will last about 30 to 45 minutes and we hope you will enjoy it. Just before I give the floor to Sophie, I would like to shortly introduce Technobis Crystallization Systems to the ones to you that you do not know crystallization systems. I see a lot of known names attending today, but for those that do not know Technobis Crystallization Systems, we are a Dutch company located in the north of the Netherlands, active worldwide since 2005 with more than 600 instruments sold worldwide. Our product portfolio, in our product portfolio, we have three products: Crystal Breeder, Crystal 16, and crystalline instruments for solid state research, process development, and formulation. Shortly on our instruments, I think a lot of you, many of you already have seen or worked with the Crystal 16 or the crystalline, even the Crystal Breeder instrument. If after this webinar you think that you would like to test one of our instruments in your lab, please let us know. Contact us directly via the website or send us an email at info@crystallizationsystems.com, and we will be happy to bring an instrument, a demo instrument, to perform a demonstration in your lab. Just before we go further, Crystal 16, the version 3 of it, newly launched just before the summer, is your instrument, remember, that helps you to determine solubility, temperature dependent solubility curves, metastable zone with the new function of induction time measurements, is actually quite a next level when you perform this type of instruments. Much easier, much faster. Helps you to get reliable data in a shorter time than usual. Phase diagram, solvent screening, polymorph, salt, co-crystallization, any type of solid state type of screening is available on this instrument, on the Crystal 16. Crystalline instrument with the camera technology and Raman, the camera technology that works exactly like a microscope. And the Raman helps you to develop your process or optimize your process. It is your instrument for particle engineering. And the Crystal Breeder, also for this one, this summer we had a few gadgets newly launched, the evaporation and the vapor diffusion setups. I like to call them gadgets. This really enables you to perform controlled crystallization, diverse crystallization methods like vapor diffusion, sublimation, evaporation, you name it. It helps you to grow single crystals with minimum amount of material in a reliable way. And this was about Technobis Crystallization Systems. And now it's time to introduce Sophie Janbon, our presenter today. Sophie did her PhD in the group of Professor Roger Davey back in, sorry, I hope I'm not messing up my screen. From the University of Manchester, the well-known Professor Roger Davey. In 2007, she joined AstraZeneca shortly after her PhD, a senior crystallization scientist. Bear with me, I have only one monitor and I'm trying to fiddle between different screens. Her main activities were to develop robust crystallization processes for API to provide suitable crystalline forms and particle properties for downstream processes, including efficient isolation, drying, formulation development, and drug product manufacture. Since 2017, she was promoted to Associate Principal Scientist in Crystallization Science. And since 2018, she is a team manager of early development phase. Her team focuses on biocatalysis, crystallization science, material particle science, and process engineering. Sophie, thank you very much once again for being able to present, to bring this webinar to our audience today. As always, a pleasure to work with you and to have your work presented. I am going to give the floor to you now.
Hi, everybody. So thank you very much, Carmen, and the rest of the Technobis team for letting me speak today and present on my experience of working in the crystallization science in the early phase. So I will cover mainly here phase 1 and 2, and basically the challenges for API crystallization processes in that clinical phases aspect. And I just want to say that this is my experience that I'm sharing with you, and I hope you will see that it's a bit of a mix of basically some kind of what's going on in the pharma to my, or at least in AstraZeneca to my experience, and some technical project as well, a bit more the impact on what's basically going on in terms of how we develop crystallization processes. So I will remove my camera just because then I get a bit more internet bandwidth, and hopefully that's fine for everyone, and then I will keep on talking. So is that all right? I'll remove my camera and you can still see the presentation, right? Okay, so I will start. So I will try to not waffle too much because I know that we've got 30 minutes, 45 minutes, and you want time for questions. So please, maybe 5 or 10 minutes before the hour, let me know because I can basically speak a lot. So that's why I often put an outline just to make sure I keep track of, and basically I keep on talking through my presentation in a very structured and not too waffly way. So I will cover basically what crystallization science is, what are the challenges I basically want to cover in the clinical phases 1 and 2, what kind of scientific approaches we're using in the early phase to tackle these challenges. I'm saying in the early phase, but obviously this is used across the entire life cycle of the medicines. I will show you some project example, and then I will conclude. So basically, I think we all know what crystallization science is, but there is always a picture which comes to my mind, and I like presenting that in conferences or webinars, just because it's sometimes how I feel when I talk to chemists or process scientists in general. I feel like a bit as I'm a witch, like I'm creating a crystal. Wow. Obviously, this is not the case. I'm not creating something new. We all know that it's science. And this science, where it is located in AstraZeneca, or how I basically have been experiencing it in in my career is basically being at the interface of drug substance and drug product, basically at the end of the chemical synthesis and at the start of the formulation development. So I'm very focused basically on delivering the right form, and in this talk I will focus mainly and only on crystalline. I'm very focused also on right particle. Is it the particle properties appropriate for the formulation platform we are going ahead with? I'm very focused also on the right purity profile, just for the patient safety. This is very important. And then also right throughput and sustainability and yield, just to make sure that our process are basically efficient and we get the most out of it. All the talk also will focus on batch mode and will not touch too much, not touch at all in anything on continuous, but basically this is where crystallization science do land in the drug development within my experience. So crystallization is basically getting a solid out of a solution, and we've got that is a non-exhaustive list of processes we can go with, and there is action which should not happen, but never mind. So we've got cooling and anti-solvent process A and B, which are the most common ones for free molecules in the pharma industry. Cooling with basically by using temperature, you crystallize your compound. Anti-solvent by using an anti-solvent, adding to a solution of the API. And then you've got also in terms of changing basically more reactive, you've got pH change when you change the pH of the solution of in which the API is in solution, maybe especially for free form as maybe ionized form, so you get it as a non-ionized and crystallize out. In salt formation, you get it as a free form, and by changing the pH, you basically with a counter-ion, you're basically forming a new chemical entities. And then you've got more way where you remove the solvent through a natural process like evaporative, or through more engineering aspect through distillation. And that's how you basically can crystallize your API of interest. But you need to choose the process you want depending on what you need to make. So it's very important to understand what you're actually trying to crystallize. Is it salt, a co-crystal? Is it free form? And that's really important and will help you to define which is the best way to get it as a crystal out. So I think that's just to a first conclusion about that chapter is the crystallization science is, to my experience, at the interface of drug substance and drug product, focus on form, particle, yield, throughput, and sustainability, as well as purity. And I've shown you many ways to initiate and control crystallization, and we'll focus mainly on batch mode. So let's focus now a bit more on the challenges in the early phase. So first of all, just to give you a bit of an overview of the life cycle of the medicines, basically, I'm just going to remove that because I can't see. There we go. Basically, AstraZeneca spanned the entire life cycle of the medicine from research development to manufacturing and supply, and the global commercialization for primary care and specialty care medicines. So can I have no? Oh yes, I think I can do. I hope you can see my pointer. There we go. That's better. So where I am actually, my team and I are basically focusing our work is around phase 1 and phase 2. But you can see quite a lot is already being done in terms of finding the potential medicines of the API, some preclinical studies, and it's where we'll pick up the project and we basically develop the process for phase 1 and phase 2, and then we give that new process for phase 3. So it's the late stage in our organization taking care of that, and all the way to commercial. So we will focus mainly the talk on this phase 1 and phase 2 part. So what are the challenges? I've been thinking about that for a while, and I think there is four main challenges in the early phase. It's basically materials, representativeness of those materials, knowledge, and time. And I'm just going to through all of them individually and give you the reason why. So I think time. Let's think 10 years ago, to bring a molecule to market was 10 to 15 years. Nowadays, we've got in our pipeline, in terms of small molecule, Tagrisso or osimertinib, which basically took 3 years to go to a commercial, which is non-precedented, brilliant in terms of AstraZeneca drug life. And COVID-19, less than 2 years. So you can see that we are actually being more faster to make those important medicines available to patients. And that is really key, because a patient cannot wait sometimes 10, 15 years to get that valuable medicine. So in some ways, ethically, we've got to be more efficient. And the impact basically of that, it's we've got faster timeline, shorter window of opportunity. So we've got to be quite proactive, and we've got to be very strong in terms of prioritization and choosing our most technical challenges. So there are aspects of rapid design of processes to deliver those invaluable medicine to patients in clinical trial. So really, that's important. The time have now changed. That's something to really keep mind of. In terms of knowledge, when we take a project from critical phases, let's be clear, we've got not much information to literally nothing. So we've got many questions unanswered. Sometimes is it crystalline? Is it amorphous? Is the crystalline phase we've got is the most thermodynamic stable form under manufacturing storage condition? Is there solvate hydrate? Have we got good biopharma properties, good chemical stability? Can we make it robustly and consistently? Can we make it the right thing? And the particle properties, have we got any information? Any impact on solvent and purities? Basically, we have to start from a blank canvas most of the time. So when we take a project from early phase, we've got to gather a lot of data in a very short period. So sometimes one experiment can span a lot of data in one go, and we've got to be very clever in our design of experiments. If we look at the third challenge, which is around materials, when you take a project from preclinical, I can tell you that we are definitively scrapping barrels. There's not much material available for meaningful crystallization work. So we've got to be very smart with the way we design our experiments. So often we get 5 to 10 grams available for the first GMP scale-up development phase, when we've got to develop a process which will basically be above a kilogram of process. So 5 to 10 grams to try to understand what are the risks at scale can be quite challenging. And often we have no GMP materials. So we are very constrained in terms of seeding protocol, because we can't put non-GMP material in a GMP batch. We've got to be very quality conscious about that. So we've got to be very smart and very key and very focusing on what's critical for the delivery of these investigational medicines. Finally, the fourth challenge is basically representativeness. Representativeness, I don't know if it's a word, but I can tell you I'm using it a lot. So there is lots of talk about, is it going to be representative? Is it going to be representative? And you wonder, representative of what? And often people think about more commercial or the next clinical batch. And you're thinking, when you start from a preclinical sample, is not being made in an optimal way, so it cannot be completely sometimes pure. So you've got to understand that it might have different impurities as well as what you're going to make in the future. So the main thing here is you're never going to change that. It's got to be risk assessed on case by case and understand basically the impact also it can have on the crystallization behaviors. And sometimes it can be a red herring, sometimes it's not. It's really important to understand how much the quality of your material may have an impact on the crystallization and form control as well as particle properties. And obviously, purity control, process usability delivery is definitely impacted also by representativeness. So that's something which we've got to risk assess on a constant basis, and we've got to live with. So I give you a feel of basically the four main challenges we've got in the early phase. Now, what are the approaches or the scientific approaches we've come up with to tackle those challenges? So back to the challenges, I will focus on materials, representativeness, knowledge, and time. So let's start with time again. I think you've seen the mega trend is bringing faster medicine to market, and that's important. And I think AstraZeneca has really transformed that in terms of R&D productivity. Just as an example, we used to have drug nomination to phase 3 completion in 2005-2010 only 4%, and now we managed to bring it to 19% from 2012-2016 years. So it's a pretty big achievement. And by doing so, we basically improve the prioritization of our process based on technical challenges. So you can look at, you can read two papers, which are the information on the left side of that slide, around the AZ5R framework. So really bringing the right molecules to a phase 1 clinical by focusing on the right target, the right tissue, the right safety, the right patient, and the right commercial. So you can actually have a bit more detail by reading those articles. Very good. In terms of strong prioritization on a local basis, we also work quite tightly within my team to make sure that we focus on the most complex projects and those which needs less, which are less complex. We go a bit more simple in terms of developing process, which are just okay, or basically we cannot work and we've got to give that to other people. So, and we tend to focus also on fit for purpose processes to make sure that those are delivered to our patients. So we have to focus on a limited set of experiments. In terms of knowledge, I think the mega trend we all know, it's all about digitalization, data science, and AI machine learning. And I think that's brilliant because it means that we can basically now take old learning and use it for new projects, which is really, really good. And now we can do it in a more consistent, structured manner. And we can do it using our new grid ELN. So basically, it's a new approach to write to our electronic lab book and enabling basically data to be stored in a way we can search them back. So that's really good in terms of capturing the knowledge and using it for the future. There is also prediction. We do a lot of work, and that can start from molecule to batch size. So we can do crystal structure prediction, which is CSP. So it's basically we've improved our force field and we understand a bit more the theoretical landscape of a crystalline foam. We've got also knowledge-based approach to understand our crystal structure using basically the composite crystallography data center tools. That's a strong collaboration. We've been involved with CCDC since 2008. But there's lots of other things that CCDC is doing, and also in the space of particles. So I will have a look at their webpage. And also you've got more to predict what's going to happen to your batch or to your process in terms of mixing and other aspects you want to look at into your batch. You've got DynoChem, computer fluid dynamic. You've got also GPROM, and the latest I've heard of is MixIT to look at the mixing. So that is basically gathering quickly knowledge to really make a better decision to your processes and your development. In terms of materials, I think the mega trend now is miniaturization. Can we reduce the size of a reactor, make things at small scale, get information quickly at small scales, and basically recycle materials as much as practically possible. So there is lots of things going on externally and internally. From an external outreach, you can look at, for example, the University of Newcastle with Indicatrix. So from Dr. Michael Hall, so they looked at nanoscale experiment, encapsulated nanodroplet crystallization technology. So it gives you quite quickly, you can get polymer screen, single crystal data, and using very limited material. So that's one example. The other example, as Carmen nicely showed, is you've got also the crystalline, crystal 16, and crystal breeder, which are different equipment developed by Technobis, which enable you actually to develop processes. So within AZ, we use crystal 16 basically to measure solubility, develop solubility curve, and metastable zone width. And then with crystalline, we develop sometimes proof of concept of crystallization process. And then once we've got a bit of an understanding of how the process and the processor outline should look like, we try to then scale it up a bit more. But with very limited material, we can get quite a lot out of those instruments. The crystal breeder is more for us a single crystal generator, and that's used more with our solid state partner. And then the representativeness. I think it's the mega trend here is acknowledging that it always was, will, and will be a comprehensive risk assessment on how representative the API attributes will be from one batch to another, from one phase to another, and up to clinical. So the main thing is to really follow the ICH guideline, which gives you a very good framework for this type of critical activity. It's collecting as much data as possible. As I said, you've got now different framework to collect data, and it's individual for later development phases. In terms of how these impurities and the representativeness of material will have an impact on the physical attributes, and most particularly the API in terms of particle size, then you can develop a particle map. So it's like a PCA map where you look at how, in terms of particle properties, your batches are basically similar or different. So that gives you basically like an idea of how your particles are basically changing through different deliveries. And obviously, you've got process analytical technology to enable you to collect as much data as possible on an experiment work. So you've got Technobis, because by definition, through turbidity and through cameras and Raman, you can really look at what's going on in your batch. But you've got also metal Toledo probes, Blaze matrix probes. So you've got also lots of other tools and spectroscopy and vibrational method to really understand what's going on in your process while you're running your process, and try to see if there is something else you can do to improve for the next, or control and make it more robust. So that gives you basically an overview of the 4 challenges and some scientific approaches to work with those challenges, and to basically make those invaluable medicines to basically be delivered to our clinical phases and our patients. So now let's focus a bit on the project example. So I will show you 4 examples, which I might have shown you, or some of you have seen maybe over of my presentation. I like those examples because they are very indicative of the new scientific environment we are in, but science progress all the time, and there's so much more we can do. So here we come. So let's start with basically compound 1. So compound 1 is nice because I can show you the molecular structure. We know so far that this molecule has got two anhydrous forms, form A and form S, but we have found almost 19 different forms observed so far through polymorph screening. So it's a very rich polymorph landscape. So by being so rich, it basically highlights a risk for us. It's like, which first of all, between form A and form S, and over form which are of relevance in the 19 form observed, which one is the most stable under manufacturing and storage condition? And just to put it there, the anhydrous form are the more relevant for manufacturing storage conditions. So it's just understanding between form A and form S. And the main risk is basically in our head, if there is so rich polymorph landscape, is there a risk for an additional solid form which could be more stable? And that's where it's really important to get that, or to capture that knowledge quite early on. So this is basically, I'm going to show you an example of how we use crystal structure data to support drug development in AZ, and structural assessment, risk assessment, and knowledge-based prediction. So let's go to the next slide. So basically, this is the two crystal structure of form S on the left side, form A on the right side. Form S has got 1 molecular conformation per unit cell, and form A has got 2 molecular conformation per unit cell. And I've shown just the one conformation for form A, but it's equivalent in terms of a response on that, or using that tools on both. So the tools is called full interaction mapping. So that's why we call it FIMS. I have focused this basically study on just 1 molecular conformation, which you can see here. That is form S molecule, which is basically the API, compound 1 in form S structure, and compound 1 in form A structure, which is just 1 molecular conformation for this one. And the main thing I want to highlight here is that this molecule, which contain 8 acceptor and 3 donors, are still exhibiting just one hydrogen bond, one only single one intermolecular hydrogen bond. So basically, there is something else holding the crystal structure. So here, the full interaction mapping is basically looking at the molecular conformation and then generating, asking the crystal structure database, which contains now almost 1 million structure and is curated by CCDC, to look at the certain part of that compound 1 and see where most likely, geometrically speaking, the hydrogen bond acceptor, donor, and then also the aromatics will be along that conformation. So you can see in red is basically the acceptors, in blue are the donors, and then in amber is basically all the dispersive interaction. You are most likely to see them using that molecular conformation. So it seems that in 4S you've got the hydrogen bond here from that hydrogen from this ring going straight into this cloud where the most likely acceptor is, and is the same for this secondary for this nitrogen. Is it primary? I think it's secondary. And then the same here with the cloud around the donor, and you can see that it goes straight into the nice cloud. So optimally, basically what it's saying for 4S is you've got an optimal geometry of the hydrogen bonding because they go exactly within what is expected to be located, the donor, the acceptor in red and the donor in blue. So basically 4S seems to be pretty satisfied. When you look at 4A, you can see that this time the clouds are slightly off. So here the donor, oh my God, I'm confused. Basically the cloud is off, and here the cloud is off. So that is the acceptor, and you can see is off. You can see it's out of the cloud, and this one is also out of the cloud. So when it's out of the cloud, it means that the conformation hasn't managed to put an optimal position in which you most likely should find your donor and acceptors. So it seems that basically 4M has to come up with a concession when 4S managed to nicely match their hydrogen bond, optimally geometrically speaking. I hope I've been clear. And there is obviously looking at, because there is just one single intermolecular hydrogen bond, you can understand that there is other so-called weak interaction which must also drive the packing. So you can see that it's missing here and is off the cloud when it should be within the cloud, like within 4S it was within. So 4S seems to not be optimally optimally geometrically speaking done. So if you were to look now I've shown you only the full interaction map, but if you were to look at specifics and then color code in terms of risk each specific in the crystal structure of 4A and 4S, then you start really building a picture in which 4A looks less strong and show much more non-optimal features compared to 4S. So, for example, the Z prime for 4A is 2, when for S is 1. You've got in 4A a disordered structure. The 2D structure contains, as I said to you, 3 donors and acceptors. In terms of edge bond propensity, we basically have still only 1 donor and acceptor in both. But for 4S, you've got the highest propensity of the one actually existing. In terms of geometrically speaking, how they are in the crystal structure, they are geometrically non-optimal in 4A, as we've seen from the full interaction maps, but is a lot more optimal in 4S. You've got another tool from the CCDC or Mercury software, which is called aromatic interaction, and does show basically that 4S has a high interaction in terms of aromatics compared to 4A. there is no voids, and the molecular conformation is basically both non-optimal. So when you look at that picture, you realize Formes seems to be more usual than for me, but the risk of another form which could have a stronger or better propensity for hydrogen bond may still exist. So because we know that Formes appear more optimal and usual than forming, we basically focus our attention to develop a process to make Formes. And in the background, we also work a bit more to review the polymer script and making sure that it was done with a higher quality sample and we haven't missed any key experiments. We also did some crystal structure prediction work to see if there was any other possibility more stable than Formes. So that's really driving basically our risk assessment. So that was an example with compound 1, and I just want to show you another example with compound 2. So compound 2 is still a free form. We've got one relevant anhydrous crystalline solid form, which is Forme, and which exhibits also a few hydrates. All the work we've shown so far said that Forme appears the most stable, thermodynamically stable form compared to also the hydrates. We don't know any other anhydrous form. But when we review the crystal structure features and we basically risk assess those in terms of space group Z prime, H-bond propensity, full interaction maps, aromatics, VOD, and molecular conformation, you can see there's a lot of them there coming out. So that kind of highlights some kind of risk, especially around the Z prime equal 3, when you basically have 3 molecular conformation in the unit cell, which is pretty rare for what we call a thermodynamically more stable form. But that doesn't mean it's not possible. It's just for us to keep an eye on, okay, maybe we need to do a bit more work to confirm that and to give more reassurance. So as I said, Forme does not appear optimal and exhibits some unusual crystal feature, but unusual doesn't mean it's not. It's basically that. It's just for us to kick off the right activity to make sure we can reduce we understand and can reduce the risks. So we basically initiated the polymer screen as early as practically possible to ensure that that form was the one to go ahead with. And to not develop a lot of, basically the main thing is to make sure we're not developing a process which is not actually making the right form and not good for our patients. So that's why we kicked off the polymer screen early on. So now let's go to compound 3. So this time we move away from free form and we are going to a salt through a reactive crystallization process. Basically, compound 3 is made of 1 API, V2 sodium, and 6 molecules of water. So it's quite rich in water. During production 1, basically what's happened is we deliver primarily single particles, and from our particle size distribution method, we got a D90 around 50 and 55 microns. But during production 2, what happened is we actually delivered larger particle size than our clinical precedents. And basically the D90 this time was 75 microns. And what we observe is basically that new batch was making agglomerates. So the main thing is, 2 things is, do we understand how these agglomerates have been formed? And again, is there any kind of clue from the crystal structure that could indicate us to know why it's basically forming those agglomerates, and can we reduce the formation of those agglomerates? So when you look at the optical microscope, you can see that this is basically nice plates. They seem to be sticking on the top of each other on the largest crystalline face. And when we did XRPD with perfect orientation and using crystal face identification, we realized that basically this larger face is where in red you can see the water and the sodium atoms, and in green that's where the API was. So the API is in sandwich between basically water and sodium atoms. So what is peculiar about this water sodium atoms to make that face sticky? I think that's what we wanted to understand. Is there a correlation here? So let's look at the water environment. So we've got 6 molecules of water in the structure. And we have to basically, because we are making nitrates, water is also part of the process solvent. And that's really important. So you can see in that in the right side, basically the 6 molecules of water, each in their environment. So the environment of water is defined by how they are basically interacting with their environment. So here is donor, donor, and acceptor. Here you've got a donor, donor, acceptor, acceptor, and basically they're all defined like that. There is some work which was done very early on by Amy Gillian, Noko, and all, and now you can. She's called Amy Robertson, and basically she defined several environments of water. But CCDC basically confirmed in 2018 that those environments not only are basically still relevant, even though our crystal structure database have increased massively since 2003, but the most often observed water environment is basically donor-donor acceptor and donor-donor acceptor acceptor. And all our molecules of water within that crystal structure basically exhibit those water environments. So it seems to be very optimally engaged in the crystal structure. So the main thing is basically it's a very tight system in which water molecules are nicely bonded. So the question is, if we were to reduce the water in the process, but just enough to keep the hydrate stable and form the hydrate, would we manage to reduce the agglomeration? So that was one question which came about of that work. The second thing we looked at is basically the sodium atom environment. So basically here, the most likely optimal structure of the sodium is, as we know, basically sodium chloride, like octahedral. But in that crystal structure, basically the both sodium were only 5 times bonded. So you wonder if the sodium is not, because not completely optimally bonded, with not a lac and wants to basically bond with something else, a 6th friend. And therefore that phase become a bit more sticky. So that was basically potentially an initiation of an idea of what could happen at molecular level in the batch. So in terms of the conclusion of that structure investigation, we can define that water molecules appear optimally bonded to neighboring molecules. The sodium atoms appear actually not optimally bonded to neighboring molecules. So rapid assessment of the crystal structure provides potential understanding to what is going on in terms of agglomeration mechanisms for the sodium atoms and the water. So, so we were thinking that, as I said to you before, reducing the water might be one. We can't reduce the sodium or we can't give more to the sodium, but if we were to control the water, can we actually make that phase less sticky? So that's something we thought of doing for that project. Unfortunately, we did not manage to, because the project got stopped just at the time of that finding. But it'd be nice to confirm that. So let's go now to compound 3. Compound 3 is the most recent example. It's a free form. It's an anti-solvent process, so you can see it as also a reactive. Mixing is really important. So basically the addition of a solvent into the bulk of a batch to reduce the solubility of a targeted compound. And then that should promote supercell saturation and enable crystallization to happen. So we developed that process, and up to 20 kilogram batch, we have no problem. So you can see the nice drawing of a 20 kilogram batch reactor. Absolutely no problem of control of form. And then we make a 60 kilogram batch, so a 3-fold bigger. So I've done a drawing which enables you to see how that picturally look like, a 3-fold bigger. And then we got a generation of a mixture of forms. So we did not make a single forms, and we were wondering what happened. What did happen to that process, which was different? So the fault may be different, but the main thing here is the answer is what was the cause and how can we make the next batch? Because we had another 60 kilogram batch to make a success. So when looking at the process, we are thinking when you go by 3-fold, then when you add the seed, they might not disperse and mix as well as if it was a 20 kilogram batch. So that was maybe one cause analysis, we're thinking that might be the reason. So what we did is we took the tools of DynoChem and scaled down from 60 kilogram to 1 liter Optimax reactor. So this for time is a 400-fold smaller. So you can't see it, but here is basically the Optimax. So in a picture way, it's very tiny. And we use a liquid mixing model in DynoChem just to see if we can make basically a mixing scale down. And what we did is we did a design of experiments. So we decided to do high mixing and then pro mixing. So by changing the agitation speed, and we decided to do low seed loading and high seed loading. And that was basically, we came up with that small design of experiment of 4 experiments, changing only the seed and the mixing. And that was the only thing we could actually change for that next batch. So we decided to focus on what we can control and do. So when we started experiment 1, which was basically the worst case scenario, low seed loading and low mixing, we basically saw using Blaze matrix. So it's basically a path probe which we put in the reactor. We saw lovely bubble. So actually that's an oiling out. And then later on, at the end of the process, we still saw those lovely bubble. And then over the weekend, those lovely bubbles crash out, and you can see the material at the end of at the bottom of the reactor, and basically covering completely any kind of baffle present in the batch. So the crystallization was very rapid, and when we looked at the form of that solid, it was basically a mixture form. So we observe an oil and we observe a mixture form and a no control of crystallization for low mixing and low seed loading. So when we went to this time low seed but high mixing, so in this part of the design of experiments, we observed no oiling out, so 1 gain. Yes, no oiling out, that's great. The crystallization rule was slow, but it's ended up being slow with a non-desired form. So actually we ended up at the end with a mixture form. So low seed and high mixing, we avoid the oiling, but we're not avoiding the control of form. Sorry, we're not avoiding the fact that we're not controlling the form. So when we went this time with high seed and high mixing, again, no visible oiling out, so things seems to be okay. And this time we got only desired form we wanted. So in this part of the design, we got green. No problem with the oiling, no problem with the control of form. So because we are quite in a tight timelines, we decided to stop here and basically say to the project to go to the basically high seed loading, high mixing. So it was great to use DynoChem to input to our lab work to facilitate the understanding of the issue observed during manufacture. Basically, due to timeline, we definitively focus our work only on the high seed loading and high mixing, and we thought that having a low seed and a low mixing and a high seed would not enable the seed to be nicely dispersed. So we will also go to a problem of form control. So we thought best to just focus on the high seed and high mixing. And when we recommended that, we basically managed to successfully deliver in one go at 60 kilograms the desired form. So that was a great use of PAT and DynoChem to really basically deliver the right form for that project for the next batch. So that was a nice story for compound 4. And now I'm just going to conclude. So first of all, I hope that I show you that crystallization has got a lot to do, many things to deliver. Understanding is the first one, especially in the early phase. We've got to get more grips and to really make sure that if we understand what can we control, what can we make sure is going to be the right thing to go to our drug product colleagues and our patients. In early phase, the challenge is definitely to deliver the right form. Particles is definitely critical, but we're still capturing the knowledge on particle, delivering the form to get the right biopharmaceutical element is often key. As I've shown you, current challenges in the early phase is definitely time, knowledge, materials, and representativeness. I hope you've got now an appreciation of the many different approaches to manage these overall challenges in the early phase. Prediction definitively for me is really key and has to work in partnership with experimental, and I hope I show you that in those examples. As I haven't touched upon continuous, I think continuous is a way to actually enable faster delivery of our actual drug to market, but it's still a process to deliver. So that's something I am very keen and passionate about, but batch mode is still fun to work with. So there is a place for everything, but it's important to understand all technology. It's like a toolbox. You need to choose the right platform for the right compound. Finally, I will acknowledge, I think I've worked with so many colleagues in AstraZeneca, and I've enjoyed my time with all of them, past, present, and hopefully future. All have been great scientists, and they made me enjoy my job. I would like to thank very much the Reverend Richie Gillian for the picture of the witches. This is my favorite picture, which made me feel, as I said, a bit, I don't know, some kind of, sometimes I feel like process scientists look at it this way. Anyway, and thank you to you all for listening to that. And if you have any questions, please let me know, and hopefully I will answer them.
Thank you very much, Sophie. I am. Let's put the camera on. We do have a few questions for you. I will try to, if you can leave your slides up, because maybe you will need them, Sophie. Yes, perfect. So I will try to read now some of the questions. One of the questions is from Karina Wang. Hi, Sophie. For the very small scale reactor, like 1 milliliter, do you use inline or offline analysis to determine the formation of crystals, of a crystal? One second. How do you determine if the crystal is formed instead of amorphous reprecipitation? Thank you.
So in 1 minute, so you're talking about 1 minute scale, Carmen?
Yes, 1 milliliter, yeah, scale.
Okay, so for 1 milliliter scale, I mean, first, the crystal 16, which I tend to use, but other things, visual anyway, you will see that there is a solid. For me, a solid is just a solid. You still need X-ray diffraction to confirm that it's crystalline, or optical microscopy to look at birefringence. So I will always take a sample of your slurry and put it under optical microscope if you don't want to disturb or isolate your 1 ml vessel, and look at if there is any birefringence. And that will give you quickly if you're making what you expect, the right stuff. I hope it's crystalline. Or if basically amorphous precipitation is what you want, does it show no birefringence? And if you want to know more, I think X-rays is the second aspect.
Thank you. 2 questions from Olainka. It's good to see you here, Olainka. Form A and Form S. Do you only use modeling to confirm the thermodynamic stable form? Is one of the questions.
So do you use?
Do you only use modeling to confirm the thermodynamic stable form?
No, we don't use only modeling. Modeling was a nice example because in this case it's just highlighted that. So we don't do only modeling because it's important to confirm that at any temperature the form you want is the most stable form. So you need to understand that manufacturing condition. The prediction or the modeling aspect was to risk assess if it's most likely to find another more stable form. So if form S was completely green in terms of risk, I would have said, okay, I think it's low risk to find a more stable form than form S. But in this case, there was some amber, and that kicked off additional activity to make sure we confirm that this is the right thing, and there is nothing else around. So it's really probing more. So the prediction, the modeling is only to inform how unusual or usual crystal structure is, and that's basically support our polymer screen, and the experimental has always to be done.
Okay, and his second question was if experimental, then to describe the what, but I think you already covered exactly what he was asking. Yes. Another question from Michaela Pop, a CRO, Terra Crystal. What method software do you use for CSV?
What software?
Yes.
I am not the expert on that one. It's my solid state colleagues. I think they use I don't want to answer something stupid. So what I can do for that is I will confirm that with my colleague and then bring back the answer to you, Carmen, and then you can reply to.
Yes, or I can share this with you directly after all the questions, Sophie. So then you will see exactly.
It's not expertise, so I prefer making sure I'm not saying something stupid which is not going to help anyone.
No worries, perfect. Another question from, and they are coming. More questions from Ankur. How do you decide on the rate of solvent addition into anti-solvent? Are there some designs or design of experiments, most probably what he is meaning, which could improve the contact time? What mixing parameters are important during the crystallization?
So I think that's a very good question, and I think that's basically trial and error at the moment still. So you've got to start somewhere, and sometimes basically sometimes is kind of detected to you because of the manufacturing equipment and setup you will put the process in. So if the process is using a certain pump or certain things, then you will have a certain limitation from that large scale equipment. So you've got to understand that. And then in the lab, you need to use, as you just rightly said, a mixture of design of experiment and that, and looking at critical process parameter in your vessel for the process itself. And in terms of mixing, it can be quite a lot because your ampulla design, the speed of agitation, your reactor design, where do you add the anti-solvent or the other element of you want to add. All that will have a massive impact. Also the size of your droplets going into your reactor. So it's actually not easy to understand what is the optimal setup. It's definitely a drug development activity, experimental work to be done. With now, obviously, DynoChem, CLT, and GPON, we can get more understanding what is going together with experimental. And where you're going to put, understanding where you're going to put your process into.
Okay, perfect. Thank you, Sophie. Another question from Julian. To select the most appropriate physical form, are you also looking at bioavailability aspects to save some time, money? Example given kinetics of dissolution in biorelevant media?
That is definitely the first primary driver. That is also biopharmaceutics. Biopharmaceutics will always win, and then we have to look also at the solid state properties. Together, then we've got to understand if it's something which it's basically stable, has got good properties, it's handleable, but is also the right dissolution and the right exposure for the patient. If we don't get that first, then we cannot keep it anyway. So I think biopharmaceuticals is critical. But the main thing is, as I said to the people I work with biopharmaceutical and state, is that if I can't make it, it's not going to happen. So it's really important in terms of form selection for every partner to sit down and decide the best for the product.
Okay, in the meantime, I see an answer from one of your colleagues, Stan Nielsen, Leo.
Yeah.
Yes, that he is jumping up here to answer the question on the CSP. And I do see another question related to the CSP software from Peter Doyle. Thank you, Peter. I will share everything with Sophie, and then she can also contact you with direct answers or her colleagues. So Stan, thank you for jumping in here. He's saying we use an internal developed force field in connection to GRACE software developed by Avantgarde Simulations. We have developed a quick approach allowing us to use this in early development phase. Thank you very much, Stan.
Yes, thank you very much, Stan, because Stan is our crystal structure prediction guru in so it's good, but he was online and he can reply. Thank you.
Super, thank you both. And we have one more question related to induction time measurements. And the question is how to identify induction time and completion of crystallization if change in case of polymorphism. So in case of changing the polymorphic form during the crystallization.
So I think the main thing is if you are in the phase of crystallizing your API, you've done some solubility work, and you've done that solubility work more specifically on the more stable form at different temperatures. You've got the solubility curve. So you've got two ways to prove that your crystallization is over. Is either you accept basically your path data, which you like an APRF, you train now plateau and you know that nothing is coming out, and you know that your crystallization is over. But it's not very accurate. Like sometimes it's just because there is no driver anymore. So I think the most to liquors is basically taking a sample of your slurry at the end of your process, taking your liquors, measuring the concentration of your API in liquors, and comparing with your solubility to see how far you are from your equilibrium. Obviously, now we're talking about polymorphism. It's good to really check that you are crystallizing your process in the right form. So take a slurry, filter it, get a bit of a solid, put it on XRPD, just to check the right form you made. Check the liquors to check how far you are from the solubility, or are you basically meeting the solubility. And then PAT will also tell you if the process has now achieved basic steady state. And so that's a few ways to really make sure you've got the right process when you are at the end.
Perfect. Thank you, Sophie. One last question, just popped up. Is from Emily. She's saying, thank you for the talk. I think all examples were polymorphs, but I was wondering, how do you screen for crystal habit? Mainly microscopy?
So I think that's a great question. That's something we are doing. So solvent, lots of things can affect morphology. And it's really important. What we do nowadays is we actually doing morphology screening. So we do API, and then we let it evaporate, and then we look at how the shape may change. But you've got also engineering ways to basically engineer your particle shape. So recently I work on a process where I use wet mill, and that enables you to tune the particle shape and the particle size. So you've got also engineering solution to control the particles size and shape. We do screening, but we also do proper engineering on particles to sometimes we manage really straight away from the process. And we do the right process for particle size reduction to control the size. So we've got lots of ways to control it, either perfectly or because the API is just giving it the right forms, the right shape and size straight away. And that's how we do it. But yes, it's good to do a morphology screening, we call it. It's something we are trying to do more and more, I think, in the globally in the industry. And then you've got also prediction tools to look at morphology and different softwares now you can do that. And also lots of things is quite cool nowadays is looking at the chemistry on each surfaces and the impact it can have with different molecules, excipient and so on, so, or solvents. So lots going on in that space, definitely.
Yes. Perfect. Thank you very much, Sophie. I think this is it. We have covered most of the questions. As I promised earlier, I will share everything with you. And if it's something to pick up, then we will contact back the attendees and answer the questions. As always, a pleasure having you here and presenting your science. Really insightful. Thank you very much once again. Hope to see you soon. For the attendees, they are still online. We are coming back with a webinar end of September. For those located in Boston or New Jersey area, we will come physically back in Boston and New Jersey with 2 workshops end of November, and at the same time, end of November, beginning of December, we will be in London with a workshop, one of the Lunch and Learn Technobis available face-to-face finally after the pandemic. Thank you very much all. Thank you, Sophie, and have a good afternoon or evening.
Bye.
Bye.